It's a great pleasure to be here. I've been working at NYU. I keep wanting to say Poly, although we've now renamed ourselves just recently to Tandon due to a gift that we received. So at Tandon, for years now, I've passed by St. Francis a number of times, never had a chance to enter the building. So thank you very much for the invitation. I'm glad to meet some of the faculty and all the students. I've tried to tailor the talk towards students, so please don't hesitate to ask questions. If you have any while I talk, no need to wait until the end of the presentation. We work in area of, the way I think of it is biointerfacial engineering, and one application is biosensors, and that's what I'll focus on today. Some of the things that we like about this area that it attracts us to it are, it combines many skills, right? So sometimes we have to work with surface chemistry, so you think about how to attach biomolecules to surfaces so you preserve their function and their activity. You want to understand how those molecules organize and some of the thermodynamic and kinetic aspects, some of the sort of physical chemistry aspects of surfaces and biomolecules at surfaces. And then also we try to develop technology. So that involves both developing methods. So how do we look at biomolecules on surfaces? How do we look at their interactions? And then also integrated chips like this. This is essentially biosensors. I'll say more about this as the talk goes on. And today I'll focus on mostly, since it's meant to be sort of technology-focused talk, I'll focus mostly on introducing biosensor concepts, kind of like in the first half of the talk. And in the second half of the talk, I'll present a couple of stories of some of the things we're doing with biosensors. So mostly focusing on methods development and the devices. But if you have any questions about some of the other areas, please ask. And if you want to know how we attach molecules to surfaces, for example, I'm happy to elaborate. So I wanted to start off with just a couple of examples of commercial devices. So this is probably one of the best known biosensors, and that's one of the several blood sugar monitoring devices. But there's others. So this device here comes from a food safety company. So you get similar type of devices where you can look, let's say, for presence of allergens or pathogens in foods. And the way many of these devices work is you apply the liquid that you want to characterize, let's say a droplet of blood, to the end of what's called a test strip. And this liquid then, through capillary forces, just like water up a porous paper, gets wicked into the test strip. The test strip is modified with some other compounds and molecules that react with the constituents that you're trying to detect, and maybe trigger a color change, maybe they trigger a conductivity change, some kind of a change in the test strip, which is then read out by the device as a signal. And that's basically what a biosensor does. So you have a biocompound of interest in a sample, and the job of the biosensor is to generate a signal from that to tell you that it's present. So it's sort of a definition of a biosensor. Now, we work mostly with biosensor concepts that are aimed to detect nucleic acids, and sort of related technology of DNA microarrays. So that's what I'll focus the talk on. To give you some of the ideas of applications that are out there commercially, and here's a list of some of the companies that market clinical as well as research focused products. So for example, you might be interested in characterizing a sequence variation in a patient that might tell you about how to treat them better. So perhaps the sequence variation means that they respond better to drug A rather than drug B for a particular condition. So sort of the concept of personalized medicine. But also, as you may know, these kinds of devices are widely used to study things like gene expression, which then lets you learn about how the programming inside a cell translates to its functioning. Which genes are on under what conditions. What happens if I do this to a cell? Which genes are going to get turned off and which get turned on. This is, I thought I would show one example. This one's actually taken from Genmark, so this company here, of what these types of devices look like. So there's usually a station that has fluidics integrated into it, and you have these cassettes typically that you insert into the workstation. And then there's some way to take a sample. In this particular device, there's a channel where the sample gets injected, and then it flows down this channel. And you'll see all these spots in the channel. Those spots are little gold electrodes. So what you can do with gold electrodes, you can carry out electrochemical reactions. So in this case, what happens is the sample flows down the channel, and every time it gets the right compound is present in the sample, it reacts with what's called a probe on the gold electrode and generates a signal. So maybe you get a hit here, here, and here, and those particular sequences were present and the other sequences were absent. I like this technology because it uses electrochemical labels, which is something that we also do in my group. This is kind of a commercial example type of things that we do in academia. So I think you are very familiar with this molecule. And you know that you take 2 of single-stranded DNA molecules to make the DNA double helix. So given just that information, I could ask the question, how do you make a biosensor from that? It's fairly straightforward. I don't know if anybody wants to take among the students wants to take a guess, but what you really need for the biosensor is the biorecognition, right? So if I have one sequence, one strand present on my device, I can use it to fish out the complementary sequence from solution. So that biorecognition event is what's really, that's the reaction that drives the biosensor device. So it's simple. You take single-stranded DNA and all the technologies all the companies that were on the prior slide. You attach the single-stranded DNA to the surface of your sensor, and then you use it to fish out the corresponding sequence from your sample. And if it's present, you get hybridization. If it isn't, you might get some less hybridization, there may be some cross-hybridization, non-specific interactions, but generally the signal is going to be less. So very simple concept to detect the presence of sequences of interest in a sample, whether it's to see if a certain mutation is present, whether it's to identify a strain of a virus, whether it's to look at viral populations, what type of viruses are present in a sample of bacteria, same type of concepts. So some of the basic considerations to make a biosensor are summarized on this slide. So we talked about specific interactions. So you need this biorecognition. And with DNA, it's fairly straightforward. You just do the Watson-Crick hybridization. That's what you need. If you're looking for something other than DNA, maybe you want to detect an antigen, a protein, or a virus. As you know, you could take antibodies. So these are molecules that are made by any type of living form that has an immune system, and they can be tailored for specificity to all sorts of different analytes, all sorts of different molecules that you may want to detect. In that case, you'd be talking about immunoassays. One area that I think the importance of which cannot be kind of overemphasized is the importance of surface chemistry. And this is why. So you're kind of taking some kind of biomolecule, DNA, an antibody, whatever it is, and you're going to put it on this inorganic surface typically, maybe glass, maybe gold electrode, maybe a polymer film. So you're going to attach it, and now you have to worry about, well, do I preserve the function of that molecule? What if the molecule has strong interaction with a solid support? It may not be interested in performing its binding function because it's already complexed with something. So you have to treat the surface in order to prevent those kinds of interactions. And you also want to prevent the possibility of your sample non-specifically adsorbing. So you don't want the sample to stick to the surface whether or not it goes through the specific interaction that you want to participate in. Like, I wouldn't want a strand of RNA, let's say, stick to the sensor even if it's not complementary to the probe molecules, the ones that have attached there to fish out just a particular sequence. So there's probably thousands of papers that deal with surface chemistry for different applications. So there's a lot that could be said about that. And I'll say a little bit about it, but not that much. And then, of course, the other big event is, or big function of a sensor is that once the binding takes place, you have to be able to see that it's really happened. So you have to be able to transduce the binding event into some kind of a signal. Again, there are many ways to do it. I'll show you some of them. A lot of times people like to break them down into, well, is my sample labeled or not labeled? So in the case of a labeled sample, it means that I've taken the native sample, I've put some kind of a label on it that helps me facilitate detection. And then when the hybridization or binding to the surface takes place, I now have the sensor look for the presence of that label. So I'm not detecting directly the molecule of interest, but I'm detecting the presence of the label. So that's labeled samples. The unlabeled samples will be the other situation where I don't have a label present, and I'm instead relying on some kind of native feature of the molecule. Maybe it's charge, maybe it's hydrophobicity, maybe it's refractive index, whatever it is, to detect its presence. So we'll see examples of both types of modes of detection. So the most common one that everybody does, and you're probably very familiar with this, is fluorescent detection. So you take your analyte, protein, DNA, whatever it is, you can modify with a fluorescent group. And then when it binds to your sensor, you'll shine light at it, and it'll emit light, because it's fluorescent, and detect that emitted light. Let's say with a photomultiplier tube or a charge coupled device, a CCD camera. So this is very common. DNA microarrays invariably function this way, with some very few exceptions. Classically, radio labeling was used. I'm sure that's something you've also heard about already. This is hard to incorporate into a biosensor, and people don't really like to work with radioactive materials, so fluorescence has really displaced radio labeling these days to a large degree. A method that is, in my opinion at least, really readily incorporatable into a biosensor are electroactive or electrochemical methods. And there's many different classes of electrochemical methods. I'd like to illustrate some basic principles of how they work. So for example, I may have a technique, like in that GenMark device that we had a few slides back, where I had the channel with the multiple electrodes in it, a technique where I detect current. So I have an amperometric technique. And I may have a label that I can attach to my sample, and it's going to be an electroactive label. And what that means is that it's something, some kind of a compound, some kind of a group, chemical group, that it can give up an electron. And hopefully, it's stable enough. So when it gives up an electron, yes, so now it's kind of oxidized, right, because it just lost an electron. But you want it to be stable against a nucleophile, because you'd like to be able to put an electron back. So you can look for the presence of the molecule over and over again. So it's reversibly oxidizable. So it can undergo reduction oxidation cycles in series. So a common label that we use is ferrocene. And you can tailor ferrocene to have different properties. We'll get to those later. OK, so how does this work? So let's say I want to induce an electron to give me a signal. So I would apply a positive potential to the electrode that's on top of the sensor. It means I'll pull some electrons out of the metal. I'll leave the surface net positively charged. And eventually the electrons, it's looking at that positive charge, is going to come across. And now I detect that current. And if I can count the electrons, I can know exactly how many molecules I have present. And from that, you know everything, because you can figure out the thermodynamics of the reaction, the kinetics, to what extent it took place. And then you can reverse the polarity of the electrode. You can go negative, and electron will hop back on. So that would be reversible amperometric type detection. That's the basic concept. So those are some ideas of how you could detect labeled samples. There's this other class of techniques that look to detect unlabeled samples. Let me start again with since we're talking about electrochemical methods, let me illustrate a label-free electrochemical technique. So maybe we can guess. You can probably guess what I'm going to tell you. So we look at the analyte here. If it's a nucleic acid, it's going to have lots and lots of negative charge because of the backbone phosphates. So now what's going to happen when I bring this negative charge down to the surface through hybridization? What's going to happen to the electrons in the metal? What are they going to want to do? They want to get pushed out, right, as the arrows indicate. So we come down, we hybridize, pushes the electrons out. But I can say, wait a minute, I'm not going to let them go out. So I'm going to apply a negative potential down here to prevent those electrons from leaving. So I'm essentially going to keep them confined. And this is a technique that's a simple illustration. All these concepts are really simple, where it's potentiometric. So you measure binding by essentially measuring the potential that you have to apply here in order to enforce current. And the more molecules bind, the more the potential you have to apply. Label-free. So in this case, you're relying, right? So the big idea is that you're relying on the native properties of the analyte itself. It's nicer in some ways, because the good thing about the label-free methods is you don't have to modify your sample, which is always good. The less messing around with it you have to do, the better. It could be in principle more reproducible. It can be faster because there's less steps. You can save some money because you don't have the costs associated with the labeling. The bad things are it could be less specific, right? So if something else that's negatively charged adsorbs here, I can't tell the difference between it. If only the sample was fluorescently labeled, I would not see the non-fluorescent background molecule that might stick to the surface. But if If it's not labeled and I just look at charge effects, it's a little bit hard to tell sometimes. Depends how pure your sample is. So there's trade-offs. Another common label-free method is based on mass sensing. And there's many ways of doing this. There's some really sophisticated methods which you may be familiar with things like cantilever. So people can nowadays make these nanometer thin beams or even micron thick. This depends on how you want to make it. And you can set them into oscillation, and when molecules absorb, it makes them heavier and the oscillation slows down. Kind of like making a pendulum on a clock heavier. The period is going to increase, it's going to become longer. Before people started working with cantilevers, there was what's called the quartz crystal microbalance, which is sort of schematically depicted here, which is a similar concept. So what you have here, you have a crystal. Typically, this would be a crystalline quartz. And crystalline quartz is piezoelectric. And that means that if I take a sliver of this crystal and I put a pair of electrodes, so these yellow rectangles are electrodes. And I can use those electrodes to apply an electrical field across the crystals. And if the material is piezoelectric, what it means is that the material is going to move. So I apply an electrical field, and that makes the crystals deform. It'll move like this. It'll kind of shear. That's kind of a shear deformation is what we call that. And I can apply that potential at different frequencies. So I can shear it, and I can hold it, or I can shear it and reverse the polarity to cause it to go back. And there's going to be a certain frequency at which this back and forth motion is optimally energized, and that's called the resonance frequency. And that resonance frequency is very sensitive to the mass of the crystal and anything that's on top of the crystal and attached to it, including any biomolecules. So it's a mass-based technique at kind of at the simplest level of description, where if binding takes place on top of this crystal, it's going to change the resonance frequency. It's going to slow it down a little bit. And I can track that with an electronic circuit. And so my signal will look something like this. So no binding, I have some resonance frequency that's characteristic of just the crystal itself, and then the molecules start stick to it, that's going to decrease. So this is a mass-based, label-free transduction technique. Okay, last example of a label-free method, and I like this one. This is something we're starting to get interested in with a colleague of mine by the name of Steve Arnold, who's in applied physics at Tandon. He's one of the pioneers in this area. It's perhaps the most sensitive optical method to detect molecules. And this is how it works. And there's tricks you have to play to make it really, really sensitive, but this is the basic essence here. So imagine I take a glass sphere. It could just be amorphous quartz, amorphous silica. Sorry, I should say amorphous silica. I can then have a way of putting photons inside this amorphous silica, and if they're in air, then what's going to happen? I can get a situation where these photons, so this is a light wave, they undergo total internal reflection because the refractive index of the silica is higher than the refractive index of the surrounding medium. The medium could be air, or it could be in water, so you could do this in a biological type aqueous environment. So I put the photons in, and the photons become trapped. And they go around and around and around. They're not perfectly trapped. It turns out there's some tunneling-like effects, but they can be trapped for a very, very long time. Now, one thing to sort of realize is that the frequencies of light that I can trap inside this particle depend on wavelength, because I have to have an integer number of wavelengths around the perimeter of this silica microcavity, they're called. If I don't have an integer number of wavelength, then what that would mean is that when the wave comes around and meets itself again, it won't be in phase. So I won't get perfect constructive interference. I'll start getting destructive interference, and it kind of just negate itself out. So there's a condition of resonance. Only certain wavelengths can accumulate to large energy densities inside this microcavity. So you have resonance. And now what happens is if a particle adsorbs to the silica cavity, the refractive index is going to change a little bit because I replace some of water or air, whatever it's in, by this particle. And if the refractive index changes, you probably know what happens, right? You probably know the refractive index inside a medium the wavelength inside the medium is given by the vacuum wavelength divided by the refractive index n of the medium. So the wavelength is going to change. So now the wavelength that gets trapped inside the silicon microwave is a little bit different, and I can sense that. There's going to be a shift. This could be a wavelength here, or this is frequency here, but equivalently you can look at changes in frequency or the wavelength of the light that gets trapped inside. And this can be done very, very sensitively, because you can resolve wavelength differences to within tens of femtometers. So very, very tiny differences, tiny enough to be able to see single molecules. So Steve's group has seen a single albumin proteins with this, and other groups have pushed it down to just 5 kilodalton limits. And the way you couple the light in, one thing I sort of omitted is sketched out here. So you just take an optical fiber, you make it really thin so that the light tends to leak out of the fiber. The fiber, by thin, meaning like something like 5 micron diameter or even thinner. So there's lots and lots of the radiation sticks out of the fiber, and then when it comes near your silica cavity, some of that starts to couple into the modes inside the cavity. And then you take a distributed feedback laser, which is sort of an affordable laser scanning, wavelength scanning laser, and you can look by scanning the wavelength, you can see which wavelengths are in resonance. So that's I wanted to say a little bit of spend time on that, because that's a very sensitive label-free method that's starting to increasingly get used for biosensing-like applications. Any questions from students so far? All right, so some other things to mention about biosensors. So why do reactions on the surface? They're complicated, right? I put a biomolecule on a surface. The surface might perturb the biomolecule. I'm going to get non-specific absorption of the analyte. Why am I bothering this on a surface? And the big advantage of surfaces is that you can really multiplex things. Now, you may need it also, like if you go to electrochemical detection, you actually have to do it on the surface of the electrode. So there may be other reasons to do it. But a really big advantage of surfaces is that you can look at not just one analyte, but at many at the same time, such as on a DNA microarray. This is a microarray that's printed by a collaborator of ours up at Columbia. And so instead of looking for just one sequence, I can look for many sequences and wherever it lights up. So this was a fluorescent detection. So here, 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 you have lots of sample present and less so of the other sequences. I want to put this slide on because it's my understanding that St. Francis recently got a microarray spider from Array. So this is one of their systems. You probably have a much newer one. This is taken from their website probably a while back. So how do you get these biomolecules on surfaces? So one way is to contact print them. So the technology here makes use of printing pins. So you have pins that have some kind of a small gap in them, which is then used to pull up by capillary force by to wick solvent with the biomolecules of interest. So here's the end of the spin, and this is after it's been filled with liquid. You then contact it to your glass slide or whatever surface you're trying to pattern, and you leave behind a little drop. And you can print an array of these biomolecules this way on a solid support. Other options that are out there are you can combine photochemistry and lithography. So you could take some kind of molecules that you put on your slide or your biochip that they can activate with light. You can deprotect them. So you activate locally by shining light just an area towards immobilization of the biomolecule of interest, and you can pattern things that way as well. And people have also taken inkjet technologies. So Agilent is a company that makes these types of supports by shooting little droplets of reagents and synthesizing in this case, the DNA of interest directly on the slide. So there's more than one way of doing this. This is one of the chemistries that we've used. I want to give you one example of how you can attach biomolecules, in this case to a aldehyde activated solid support. So aldehydes are reactive with amines. So this can work both for amine modified DNA as well as proteins, because there's amines on proteins. So this is the series of steps that one uses. Usually you want to have covalent immobilization to make sure that the molecules really stay put. So if you are just starting out, like we were a few years back, you might get things that don't look quite optimal. This is an array that we're trying to print. And we had, after printing, you go in and trying to wash off the molecules that didn't immobilize well. And in this case, we didn't passivate the surface properly. So this passivation comes back. And you get molecules all over the place. I like the way it looks. I think it looks artistic, but it was useless experimentally. And then you go through cycles of trial and error. use different solvent buffers, combinations of water with various organic solvents and ionic strengths and pHs, and figure out how to do this well. So these are pictures of printed, that the students have prepared in the lab of printed supports. Okay, so that's sort of the introduction of some of the ways of transduction, a little bit about how you can get biomolecules down on a solid support. I know there's not too much detail, hopefully, I gave you, but if there's questions, please ask. I'm happy to provide some.
How do you attach the probes onto the surface initially with your glass slide?
You mean the aldehydes or?
The DNA, the nucleic acids.
The nucleic acids. Yeah, so that's a good question. So it's actually somewhat involved, and I kind of skipped some of the important steps here. But so in our case, we buy the DNA commercially with a terminal amine. And then we, the aldehyde slides, you can get commercially, but one has to be careful. Because certain manufacturers, the repeatability of their products in our hands has not been as good as other manufacturers. So once you have the aldehyde and the amine, you'll typically go to like a slightly basic pH, maybe 8 or 9, so that you deprotonate the amine group. So you have the lone electron pair available. And the amine and the aldehyde react, and they'll make this product here, which is labile. So you can kind of go back. This is reversible. So then there's a final reduction step of that carbon-nitrogen bond to really make it permanent. And what the problem we had on the next slide, where we had the blue, it was a fluorescently modified molecule we were printing, and we saw the fluorescence all over the place, is because we didn't do this step fast enough. So they attach and then they could come off. And also what NABH4 does is it actually converts the aldehydes to alcohols. So when you do that, it kills the reactivity of the solid support as well as the ones that did attach. It makes those permanent. Yeah. So it could be gold. It could be anything that you want to put an aldehyde on. In our case, these were glass slides. Yes, yeah. So these are originally prepared by salinization. I mean, we bought ours commercially. We've actually since then switched a little bit away from aldehydes because we ended up running into some issues with the supplier we were using. So what we use now is APTs, aminopropyl trihydroxysilane, which is a salinization reaction you can put on any surface that has like silanol groups on it, SiOH groups. And then we convert those amine groups to isothiocyanate groups, and we do that in-house. And those are also amine reactive. And we found that those supports actually give us more reproducible results and even better signals sometimes. So yeah, so there's different chemistries we work with. Like if you have a really good commercial supply that can provide, these slides are reactive, so it's a bit tricky because you have a reactive slide that you get, but you don't want it to react until the very moment you want to use it. So you kind of want to have to keep it clean and everything. If you have a good source for those types of slides, then I think it's easier to just buy them, but they can be made in-house if needed.
Do you ever do it with gold? It seems like you covered the gold around the places where, like the thiol bonding, because if you used a light source, wouldn't you get a lot of reflected light off the metal? Absolutely. And that might interfere with your signal that you're looking for.
Yes, so no, you're right. We use gold primarily for the electrochemical type detection. And we've used, everything that we've done has been thiol gold based, although if you just take a DNA with a thiol on DNA and stick it to gold, we find it's not stable enough. If you have to, a lot of times these assays have to run at elevated temperatures, so you might have to be at like 40, 50, 60, even high temperature. And you may have to go for a few hours. So in our hands, the thiol gold bond is not too happy about, it's fine at room temperature, it's not so happy at these elevated temperatures. So we've come up with some workarounds where we take a thiol modified polymer. So you have a polymer that's got many, many thiol groups on it, and that sticks down really irreversibly. And then we attach the DNA to that polymer. So we have like a 2 nanometer polymer layer, and then we attach DNA to that, or whatever biomolecule you want, to that polymer. And that seems to be, you can heat it to 70 degrees for an hour or two, and it seems perfectly reversible under those conditions. Yeah, so we've ended up, I don't know, we spent several years with different chemistries. It's extremely important. It's not the, I would say, practically it's maybe one of the most important considerations in my experience. It doesn't always excite students, especially engineering students, to say, okay, now I have to go figure out the chemistry of this or that or the other thing. But it's important, and you get, this is how, it's necessary to get good quality data, absolutely. So first there was a collaboration to make CMOS biosensors. So CMOS is complementary metal oxide semiconductor. So this is the same type of device that you would make a Pentium chip with, except we're going to manufacture, instead of Pentium chips to do computing, we're going to manufacture chips that can do biosensing. So I have to work with an electrical engineer. So our collaborator was Ken Shepard at Columbia in the electrical engineering department. And his group is a circuits group, so they're very, very good at designing circuits to do almost anything that you'd like. And there were sort of two technologies that we're developing with Ken. So one was aimed at fluorescent transduction. So the idea was to take, instead of having a macroscopic fluorescence scanner type device, shrink all of that functionality into a small chip. And the other one was aimed at, and here's a picture of the chip that was fabricated. This one had just about over 2,000 light sensors, photodiodes on the chip. And the second type of chip that we worked with Ken on is an electrochemical chip, which had an array of electrodes. So these little squares here are gold electrodes. These are the working electrodes. So this is where you would attach the DNA or the protein. And then there's also counter electrodes, because as you know, in electrochemistry, you apply potential between a pair of electrodes. And then there's also a reference electrode if you're an electrochemist and you're wondering where the reference was. And that was done macroscopically. So we connected this chip to a macroscopic silver chloride reference electrode that was placed directly into the solution. I'll show you a little bit how this was assembled in a later slide. And the reason for CMOS fabrication is that if you make a lot of these, it can get relatively affordable to make these devices. Actually getting a little bit back to your comment, Robin, about the reflection and so on. So with fluorescent detection, when you have the laser on, one problem is, so you need a laser to excite the fluorescence, right? So I carry out the reaction, and now I have fluorescent labels on the surface of my sensor, and I have to detect those labels. And I do it by shining laser on them to excite the fluorescence. Now, the laser part is a problem, because the laser is very, very bright, and you might have a tough time seeing the fluorescently emitted photons, which you're trying to detect, against all that background. So what's done in a commercial array scanner or fluorescence microscope, whatever you're using, is that they use optical filters, right? So you put a filter that only transmits to the detector the photons of the wavelength that you want, and thus you block all the photons of the other wavelengths. This was a problem for the CMOS chip, because if you ask a commercial fab that makes these chips to make a filter on it, they don't really know how to do that. And you could do it post-process, but it's difficult. So we had a workaround where instead of doing things with filtering out the unwanted photons, we did things in a time-resolved fashion. So the way this chip operated is that after the laser is turned on and excites the fluorescence, you then kill the power to the laser. This is just a photodiode. I think it was like 15 bucks. So when I say laser, I don't mean like a $20,000 piece of equipment. This is like the kind of thing that could be in here. It was like 15 bucks. You turn that thing off, and you have the excited molecules. It takes some time, typically nanoseconds, could be 5, 10, 20 nanoseconds for them to emit the fluorescent photons. So we turn the laser off, and now you're sitting in the dark, except for the fluorescently emitted photons from the molecules, and you integrate that signal. So that's the way we got around having to fabricate these filters, which are typically used on these chips. And here's just an example of some data. So we took these chips, we just printed large spots. So we didn't have 2,000 different sequences. We just printed one sequence, and the spot probably covered like 100 of these photodiodes. Here's an image of that spot taken with a cooled CCD camera that was above the chip, and that's this data in the blue. And then we also read the same intensity, but with the chip electronics, so with the photodiodes itself. And you can see there's a good correlation. So the argument being is that we could replace this macroscopic detector that's typically used just with the on-chip electronics, on-chip data acquisition, on-chip analog to digital conversion, on-chip storage of the data, and then downloading off to a device or a computer or wherever one wants to process the data. And then this is the sort of the background that was measured outside. Like if you look at this right here, it's darker. There's always some fluorescence there, so that was the fluorescent background. So it's an example of a CMOS fluorescence-based biosensor. We also did an electrochemical one, and I'll show you how this one was integrated. So this is it says potentiostat on a chip. It's if you do electrochemistry, you'll be familiar with potentiostats. This has about maybe 2/3 the functionality of the potentiostats on the chip. What it doesn't have is the application of the voltage to drive the redox reaction. We're going to be pulling electrons out of a molecule to detect the molecule's presence. So that part was actually done externally, but it measures the currents and digitizes the currents and all that on chip. So you get the chip, you then mount it in this die. This is the die. There's electrical wires that have to be protected by an epoxy. So this is what a mounted chip looks like. And then here I just show you sort of the assembled device. So then the chip gets plugged into a circuit board, so you can use that to connect to the external world. And you put some kind of a container about which you can then fill with your sample of interest. And by putting molecules on these electrodes, the sequence you're trying to detect, you can then look for the presence of the corresponding sequence in the liquid sample. Oh, and the reference electrode goes right through that neck right there. That's what sets the voltage reference for the electrochemical measurement. Then you can compare. So we have, so in this case, we had labeled samples. So we had redox labels. So these were ferrocene labels. Like I mentioned earlier, when we drive the electrode positive, we have the electrons coming over. And this is what the signal looks like. So we're driving the electrode positive. This is, note the axis goes positive in this direction. So we're driving the electrons positive. And if there's no molecules, so if there's no ferrocene, we get this current. And that's because if I put pluses here, I will put down, let's say, chlorine, anions. So there's still current flowing. But there'll be like the negative ions of the salt that's present in the buffer. So it's this signal. When I have the labels, when I get to a potential that causes these electrons to come over from the label, I'll get this extra current. So as hybridization proceeds, I get buildup of signal. This is with a macroscopic potentiostat, and this is done with the chip. And you can see they're quite similar. You can integrate these areas to convert to coverages of molecules per area. So they can be very quantitative with this kind of an approach. And we've also done experiments where we've sort of had different sequences and shown that we introduce one sequence, then this electrode detects the signal, the other one doesn't. Then you introduce the other sequence of interest that's probed by this site, then you get signal there. So there's control experiments that we've done. Yes.
This is just very basic. I mean, everything you do on these surfaces depends on it being solvated. And
yes.
When I look at a chip, I think of a dry thing. But I guess you answer my question every once in a while because you talk about reservoirs and
yes.
solvents and things like that. So to keep everything properly solvated for all these reactions to be happening on this, what looks to me like a dry surface, you do what?
So in our case, like for biosensors, I always think of those as operating in real time or under a liquid environment. So you design your experiment so that you can actually keep the liquid in contact with the surface of the sensor the whole time. Yes. Yes and yes, but we do it carefully. So we have this reservoir here. So here's the chip, and we can't have water going all over the place because we get short circuits. So there's essentially a kind of a flexible sheet here that acts as a seal, as a gasket, and then we pour the liquid inside here. So we don't have a huge volume of liquid, but we can put a milliliter or a few milliliters worth of solvent right here on top of the chip. This is the chip area that's open here. And this is epoxy, so this protects the local circuitry. And then this glass vessel protects against liquid getting spilled over onto the board. So we just fill this with a few milliliters of solution. We can also put in a reference electrode in there. And that's how we confine it. Now for DNA microarrays, as you may know, those experiments are done in liquid, but it's not a sensor, so there's no active circuitry on the microarray usually. So then you dry them, and then you scan them dry. Yeah, so it can be done that way also. But with sensors, I think the idea is always to do things kind of in real time in the liquid. Let me see. OK, I have a few minutes left. So I can tell you a little bit about, maybe I'll just introduce this last section. one thing that we got interested in is the role of electrostatics. So you know that we have DNA is negatively charged. And if I make these DNA layers, I have made a surface that has lots and lots of negative charge on it. And then I'm asking it to detect negatively charged molecules. And it seemed kind of reverse to us. We said, well, can we use some other molecule that will do the same function but without the electrostatic repulsion? So we got interested in using morpholinos, which are synthetic DNA-like molecules. But if you look at the backbone, there's no negative charge. So this negative phosphate that you have in DNA is absent and is replaced with, with this backbone here, morpholin rings and these phosphodiamidate groups. Now there's, maybe I'll just give you the results from this, and if there's time or if people are interested, I can stick around and fill in some of the details. So some of the advantages of using the morpholinos are, for microarray applications, we see that they print really well. And we think it's because not being charged, they are happier to pack together, and they kind of absorb to the surface better. You can actually see the size of these spots compared to equivalent sequence by DNA spots. So the morphine spots are much larger and more uniform than the DNA spots are with typical printing conditions. This was a gene expression analysis done on a bacterium called Legionella pneumophila. This is a bacterium that causes Legionnaire's disease. So we were happy with that performance. Also now, if you have these molecules that really like to stick to the solid support, you can see, well, can I use this somehow to do detection, some kind of innovative detection approach. So one idea that we got actually turned on to by the way people use molecular beacons is to think of label-free fluorescent detection. Sounds like a contradiction of terms, but where you detect unlabeled sample by using fluorescence. And with morpholinos, we said, well, if these molecules really lie down on the solid support, what we should be able to do is modify the solid support with quencher molecules, which when they're near the fluorescent groups, they'll quench the fluorescence. So you don't get the emission that you look for. However, once this morpholino hybridizes to DNA is more water soluble, it should stand up and separate the quencher from the fluorophore, and the signal should get turned on. So we did that. So we made a microarray with morpholinos where the initial fluorescence from these fluorescent groups on the morpholino probe at the end is quenched. And then we just exposed it to unlabeled DNA and the fluorescence came in because we think of this change in solubility. I think it's a straightforward idea. You can also look at charge-based detection. So because you have no charge on the morpholinos, when they react with negatively charged DNA, all of a sudden you have much more charge at the surface. And you can detect it by measuring the conductivity of this near surface region. So when you add your DNA target, all of a sudden you get much more conductive interface locally. So you can measure that using electrochemical methods, dynamic electrochemical methods where you vary the potential at some frequency. And also, lastly, what they're really good for is they're compatible with sample manipulation schemes that rely on charge-based effects. So we have a collaborator at our Abu Dhabi campus. Anyway, he's got an engineering school that opened up, I think 4 years ago at Abu Dhabi. And they have everything under the sun to make stuff. They have every equipment you could want to fabricate things. So we have collaboration with Professor Song at NYU-AD. And what he does, he can concentrate any charged species. So you can introduce a relatively diluted sample of DNA, and you can concentrate to concentrations where you can quickly realize the surface hybridization detection without use of any enzyme. So no PCR, no in vitro transcription, no whole genome amplification, no enzyme use. But you need low ionic strength. So morpholinos were perfect for this. So what we did is with Raphael, we used this technique to concentrate the sample. The sample was fluorescent, so you can see how it accumulates in these about nanoliter volume plugs. And then we put, there's a movie here, but I'll skip that. And then we print morpholino arrays. And since morpholinos aren't charged, they'll hybridize with the DNA even in very low ionic strength. So now we can get, so now what we can do is we can concentrate our sample in just maybe 15 minutes as opposed to going for multiple hours, and get the signal in 15 minutes that otherwise we may have to wait 12 hours for, from hybridization to these morpholino spots in these channels. So I kind of apologize because it's 12:01 and I think I've gone a little over, but I do want to just some of the different elements to biosensors. We talked a little bit about surface chemistry. We're also using electrical fields to control surface hybridization reactions. I haven't spoken too much about that, but that's what that signifies. I have to put on the people who actually did the work. I'm here presenting it, but the work was done by, none of this was done by me. It was done by postdocs in my lab and by graduate students, a number of whom now graduated. And here are collaborations at Abu Dhabi, I briefly touched upon, and at Columbia University. And also, if there's more interest, there's some of our papers that deal with primarily with the subject of this talk are listed here. And there's also a number of patents that have some of the other data in them, if you're interested. And thank you very much for your attention. So quantitation, I guess one could ask of concentrations. So you have an analyte present in a sample, and you'd like to know, instead of just having a yes/no answer, yes. Is it present or is it not present? Can you actually correlate with a concentration level? So you can do both, and depends a little bit on the application. So if you're just looking for presence of a mutation, you want to know whether a certain allele is present, that's a yes/no answer. And in those cases, you usually don't go through the calibration that's required to kind of back out a concentration. But if you actually want, like if you have a gene expression experiment where you have to know concentration of analyte, well, you could do it qualitatively. More signal means more is present. Or you might have to calibrate it. I think you would have to calibrate it against some kind of a reference sample. And when they do, we don't deal with the commercial microarrays, but when they run commercial microarrays, a lot of times what's done is they spike the sample with known concentrations of a test sequence, and they'll have on the microarray probe sites that look for that sequence in particular. And so there's sort of an internal calibration built in, where you've got in your sample, you've got sequences with concentration you know because you've put them there on purpose. And you see how much signal they give you on the chip. And then you have the other sequences that you're trying to look for. And you can estimate their concentration from their signal reference to the signal from the spiked-in sequences. I don't know if that answers the question, but that's one way that it's done.
You have reproducibility there?
Yeah, the microarrays have gotten actually, they've got some bad press, if that's what you're referring to. Like, they came out in 1990s. Around, decade later, around 2000, there was a number of papers comparing microarrays to one type to other types, to PCR and other techniques, and a number of papers kind of made the claim they're not very reproducible. A lot of those problems, my understanding, have been solved through, the different manufacturers kind of, I think, through the ways that the microdata is processed. If that's done consistently, a lot of non-reproducibility goes away. And recent data that I've seen from Affymetrix, there was a gentleman, a lot of microarray companies are now a little bit on the offensive because of sequencing. Next generation sequencing is really competing with micro applications. So they take pains, the commercial companies, to present data when they can, showing how reproducible microarrays can be. And this talk, they show reproducibility that was as good as next generation sequencing. So it can be, but I think it does rest on recognizing certain limitations of the techniques and how optimally the technique has to be used.
My question was really about the cost differential between using a morpholino-based array and regular DNA probe, because protocol as a biologist to get a morpholino synthesized is a lot more expensive than if you're getting DNA synthesized, and I just wondered if you...
Yeah, so we thought about it. So we write proposals to NIH, and you have to address questions like this. So I mean, like right now, you're right. Right now, if I order a morpholino probe that's, let's say, fluorescent labeled, that ends up being, $500 for us typically, and I can get a DNA ordered for that for less. And we can't, at this point, with a pre-synthesized probe, we can't even compete against something like Affymetrix, where you don't order the probe, but they'll make it directly on the surface, because that requires very few reagents, and they can, mass produce these chips. Now, morpholino, step-by-step solid phase synthesis is not very different from the way we make DNA. So I don't see any technological hurdles really. I think somebody would have to, some company would have to invest the time to do it, but I think you could synthesize morpholino chips stepwise on a solid phase type context, same way that you can make DNA arrays. And the raw materials are not very different in price. I mean, when I contacted Gene Tools, At one point, the claim was made that they could get the raw materials if it was mass produced for less than what the raw materials are to make DNA. So I think if there was a broad enough market and it wasn't done by ordering pre-made probes, but by off-chip synthesis, like DNA markers are made and that was driven by affordability issues. Because if I have to order 100,000 DNA probes, I also need a lot of money to do that, to make an array. So the way they get around is through this combinatorial synthesis, where they make the DNA directly on the solid support. I don't see any reason why morphogenomics couldn't do that. I mean, it's not something that I would get engaged in because it's the chemistry there scares me, but I think it's doable. Yes.
Sorry. It seems like a lot of the applications for fixing a strand of DNA onto a surface is you have a single strand and it hybridizes with its complementary strand, and then you look for defects. But if you put the double-stranded DNA, and I really don't know what all goes on inside our bodies, but would it preserve its kind of windy, and could, a lot don't DNAs, and I don't know if they do it as a single strand or a double strand, but bind to foreign solutes, biotic things. Could you use that? Could you use a double-stranded DNA affixed to a surface, conformationally, hopefully not compromised, and look at interactions of small molecules, solutes, and
absolutely. So, absolutely. Very good points, all of them. So like the first point about, can you preserve the Bform duplex on a surface? So it's really hard. Like you can't really look at directly. I mean, you can't do like X-ray diffraction or something to see what the structure is. But you could, let's say, probe with small molecules. So if you know that small molecule, a lot of them have specificity for the sequences, they can go in the minor groove, let's say, and look at what the base sequence is. And you can compare the thermodynamics of a small molecule binding to the minor groove on a double-strand DNA that you put on a surface.
Half the minor groove? I don't know. So the minor groove is what? Just some conformational change?
Yeah, so like when the DNA, when the double helix winds up, you'll have, there's like there's two grooves in there that you can kind of see the bases through. So there's a major groove and a minor groove. And the small molecule compounds oftentimes will interact with the edges of the bases by accessing them through the minor groove.
So you could have a sensor maybe
for small molecules.
for small molecules with
Absolutely.
double-stranded DNA.
And so we've seen situations where we had nitropsin, which is a small molecule DNA binder, and we measured the thermodynamics of it binding to a double-stranded molecule on a surface and compared to the thermodynamics of binding to the same DNA in solution. And within our resolution, they were equivalent. But I also know of cases where you can put a double-stranded molecule on a support, and gold is notorious for this. And if I have a lot of ATs, base pairs, it turns out that A likes gold more than it likes T. So if I have a like AT sequence and I hybridize it or poly-T and I put it on gold that hasn't been passivated, that A is going to say, see you later to the T, to the poly-T, and that's just it's going to dehybridize.
All lie down?
Yeah. So the surfaces are really tricky, and like you just see that your experiment is weird, and it's really, you can't see anything because it's so few materials.
What's the size limit? How big a molecule can you bind on the surface?
So we typically work with relatively short ones, so 10 to 30 mers, but we attach genes. So we attach double-stranded luciferase genes, which I think, if I remember, are 2,000 nucleotides. There's a group in Israel that specializes in solid phase gene transcription. So the transcription is done. So they put entire genes down also, and they'll do the transcription, and then the translation is done like an in vitro type method. So you can attach really long ones. And sometimes they even use it for, like some of the biophysicists will attach even chromosomes, maybe like viral chromosomes down, and they stretch them out by flowing liquid over them, and then they can see where the proteins bind, because they're kind of ordered by this flow over. You attach one end of the chromosome down, and then you flow liquid over it to kind of cause the molecule to align with the flow. And then you can take a transcription factor or I don't know, something that whatever you want to see where it binds and look at it that way. Yeah, so different sizes. I mean, we don't have expertise with all of that, but